This is Heliox, where evidence meets empathy. Independent, moderated, timely, deep, gentle, clinical, global, and community conversations about things that matter. Breathe easy, we go deep and lightly surface the big ideas.
Welcome back, everybody, to the Deep Dive. Today we're looking at something pretty wild. It's called ultra-weak photon emission.
Ultra-weak photon emission.
Yeah, UPE for short.
Right.
Basically the idea that our bodies are giving off light,
Yeah.
but it's so faint that it's almost invisible.
Right?
And scientists are using it to learn about our health.
Exactly.
I mean, it's crazy to think that there's this whole other world happening inside of us that we can't even see.
I know, it's incredible, right? We're going to be diving into a paper from Scientific Reports where they're using UPE to track something called oxidative stress.
Oxidative stress. Yeah, I've heard of that.
Have you?
Yeah. Isn't it like when,
It's basically like rust forming on a car, but it's happening inside our bodies.
oh, right, like damage to our cells.
Exactly. And it's linked to all sorts of health problems, aging, heart disease, you name it.
So UPE is like a way to see that damage happening in real time.
That's the idea. It could give us a much earlier warning than traditional methods, which is huge for prevention and treatment.
So how did the scientists in this study actually study UPE?
Well, they used a type of white blood cell called HL60 cells.
HL60. Okay.
They're often used in research, kind of like the lab rats of the cell world.
Got it. So they're easy to work with.
Exactly. And they can be manipulated to act like different types of cells.
Oh, interesting.
In this case, they train them to act like neutrophils.
Neutrophils. Those are the...
For first responders of the immune system.
Right. The ones that rush in to fight infection.
Exactly.
So what do they do with these souped up cells?
They triggered something called respiratory burst in the cells.
Respiratory burst. What's that?
It's basically like flipping a switch that makes the cells produce a burst of reactive oxygen species, or ROS.
ROS, and those are the...
Think of them as the ammunition that neutrophils use to fight off invaders.
So they're making these cells release their weapons, basically, and then watching them with this super sensitive light detector.
Pretty much, yeah. And here's the key finding. When they triggered the respiratory burst, the UPE signal went through the roof. A huge spike indicating a ton of ROS activity.
Okay, so they're seeing more light when the cells are fighting harder.
Exactly.
But how do we know for sure it's the ROS causing the light show? Could it be something else?
Good question. To test that, they used a molecule called DPI, which blocks ROS production.
So it's like taking away the cell's weapons.
Yeah, and guess what happened when they added DPI? The UPE signal practically disappeared.
Oh, wow. So it really was the ROS causing the light show.
Exactly. They basically turned off the fireworks.
That's a pretty convincing link.
Right. And to get an even deeper understanding, they used a technique called metabolomics to see what was happening inside the cells during this whole process.
Metabolomics.
It's basically like taking a chemical fingerprint of the cells.
Okay, so they measured all these tiny molecules or metabolites inside the cells during respiratory burst.
Yep. They found that several molecules specifically linked to oxidative stress and inflammation went way up during the burst.
So this is direct evidence that the oxidative stress is causing real damage and the cell is reacting.
Exactly. It's not just a theory. We're seeing the molecular evidence thanks to UPE.
This is getting really interesting. So we've got the cells lighting up, we know ROS is involved, and now we see these specific molecules that signal damage and inflammation increasing. It's like we're watching a chain reaction unfold in real time.
You got it. And it's not just about the presence of these molecules, but the fact that they're increasing specifically during respiratory burst. That connection makes UPE an even more valuable tool for understanding oxidative stress.
This is incredible. We've gone from this faint light emitted by cells to evidence of oxidative stress and a cascade of cellular events. It's amazing how much information is hidden in that tiny signal.
Yeah, it really highlights the power of UPE as a tool.
But let's pause here for a second and take a deeper dive into those specific metabolic changes in part 2.
Sounds good to me. So those metabolic changes we were talking about.
Right, are pretty important.
Yeah, those prostaglandins and isoprostans, right? I'm still trying to wrap my head around what they actually do.
Well, think of it this way.
Okay.
Prostaglandins are like the messengers of the cell world.
Okay.
When you get an injury, for example, they're the ones that tell blood vessels to widen.
Which is why the area gets red and swollen.
Exactly. It's those prostaglandins calling for backup.
So they're like the 911 operators.
Exactly.
And what about those isoprostans?
Those are more like smoke signals.
Smoke signals.
Yeah. They form when the fats in our cell membranes get damaged by free radicals.
Oh, so the more isoprostanes we see, the more damage is happening.
Precisely.
Okay, so finding those molecules increasing during respiratory burst, that's like seeing the smoke and hearing the sirens all at once.
A perfect analogy. Means things are getting pretty intense in those cells.
And this is where UPE comes in as such a valuable tool, right? We're able to see those sirens flashing in real time.
Instead of just showing up after the damage is done.
Now, what's even more interesting is that there are many different types of these molecules, each with their own specific role.
Oh, wow. So it's not just a case of are they there or not.
Right.
It's also about figuring out which specific ones are involved.
Exactly. And that's where things get really fascinating.
Okay, I'm all ears.
So inside the cells, they found 2 specific isoprostanes increasing significantly, 8-iso-PGE2 and 8-iso-PGF2.
Okay, those names are a mouthful.
I know, right? But these are like the classic fingerprints of oxidative damage.
So those free radicals are basically attacking the cell's delicate skin.
Yeah.
And these isoprostanes are the evidence left behind.
A perfect analogy, like a microscopic crime scene.
And they also found a prostaglandin called PGE2 increasing.
Right, PGE2 is a real troublemaker.
How so?
It's known to be a very potent inflammatory mediator.
Oh, so that's where the pain and fever and stuff comes from.
Exactly.
So we're seeing evidence of the actual damage happening. And we're seeing the cell's alarm system going off all thanks to these molecules. And we can connect all of this back to that initial UPE signal.
Right, it's like the common thread running through the whole story.
Exactly. It highlights just how dynamic and complex this process is.
And how UPE might help us understand it as it unfolds.
Okay, so we've got the inside story.
Yeah.
But what about the outside? Did they find any of these molecules out there in the surrounding environment?
Yes. Actually, they found increased levels of a few different prostaglandins, including PGE2 and PGD2 floating around in the fluid where the cells were growing.
So it's not just an internal battle.
Doop.
They're calling for reinforcements from other cells nearby.
It's like a microscopic battlefield out there.
I love that analogy.
Now remember, DPI, the ROS blocker, well, it did something really unexpected here.
Oh, yeah.
It lowered the levels of those oxidative stress markers inside the cells.
Okay, that makes sense.
But it actually caused them to increase slightly outside the cells.
Wait, hold on. If you're blocking the ROS, would you expect everything to go down?
It does seem counterintuitive, right? But here's one possible explanation. It might be that DPI is interfering with how the cell normally breaks down or recycles those prostaglandins and isoprostanes.
So instead of being eliminated, they end up accumulating outside the cell.
Exactly.
Wow, so even the inhibitor is giving us clues about how this all works.
It really is like solving a puzzle one piece at a time.
This is all so fascinating, but what does it mean for us, the non-scientists?
The key takeaway is this. This research provides even more evidence that UPE isn't just some random phenomenon.
Okay.
It's directly connected to oxidative stress and to the complex chain of events that it triggers inside and outside of our cells.
And why is that important for us to know?
Because oxidative stress is implicated in so many diseases.
Like heart disease.
Cancer, Alzheimer's, Parkinson's, the list goes on.
Wow, that's a lot.
And if we can find ways to monitor and manage it effectively, it could have a major impact on our health.
And that's where UPE comes in.
Potentially. Imagine a future where a simple, non-invasive UPE measurement could tell you about your current levels of oxidative stress.
So you could make lifestyle changes or seek treatment before a serious problem develops.
Exactly.
That's pretty incredible.
It's like having a window into the cellular world.
But I'm guessing there's still a lot of research to be done.
Oh, absolutely. This was just one study using a specific type of cell.
Right.
We need more research to see how it works in different cell types, tissues, and ultimately the whole body.
So we might not have UPE-powered health monitors on our wrists just yet.
Not quite.
But this research is definitely a step in the right direction.
I agree.
But how do scientists actually measure this super faint light? That's what I'm curious about. Let's delve into the technical side of things in part 3. All right, so we've talked about the connection between UPE, oxidative stress, and all those signaling molecules.
Right.
But I'm still kind of stuck on how they actually measure this super faint light.
Yeah.
It's like trying to see a single firefly in a huge stadium.
It's a great analogy. And it does require some seriously impressive technology.
I bet. We're talking about detecting individual photons
Exactly, those tiny little packets of light.
coming from these cells.
It's mind-boggling.
So how do they do it?
Well, scientists use these incredible devices called photomultiplier tubes, or PMTs.
PMTs. That sounds like something out of Star Trek.
Haha, kind of. They work by amplifying the signal from a single photon millions of times.
Wow. So they make it strong enough to be detected.
Exactly. It's like turning up the volume on a whisper so you can hear it across a room.
That's pretty amazing.
Oh.
So I'm picturing these scientists working in complete darkness.
Well, it does need to be a very controlled environment to eliminate any background light.
Yeah, that makes sense. So they put the cells in this special PMT device.
Right.
And then what? How do they actually make sense of the UPE signal?
Good question. The PMT basically records the number of photons emitted over time,
Okay.
creating a sort of UPE profile.
And this profile tells them.
It can reveal a lot about what's going on inside the cells. For example, a sudden spike in UPE, like we saw with the respiratory burst, suggests a burst of ROS activity.
So it's not just about whether there's UPE or not. It's about the changes in the signal over time.
It's those dynamics that hold the real clues.
Now, going back to that DPI molecule, the one that blocks ROS production,
Right.
we saw that it caused a decrease in UPE.
Yeah.
So does that mean DPI could be a potential treatment for diseases caused by oxidative stress?
That's a great question, and it's something scientists are definitely exploring, but it's not quite that simple. DPI is a really useful tool for research.
Because?
Well, it specifically targets certain enzymes involved in making ROS,
Okay.
but it also has some downsides. It can be toxic in high doses,
Oh.
and it can also affect other enzymes besides the ones involved in ROS production.
So it could have unwanted side effects.
Potentially, yeah.
So for now, it's more of a research tool than a magic bullet.
I'd say so. But by studying how DPI works and its effects on UPE and those other molecules, researchers can learn a lot about the pathways involved in oxidative stress.
Which could eventually lead to better treatments.
Exactly.
So DPI is like a flashlight that helps us see the problem.
Yeah.
But we still need to find the right tools to fix it.
A great way to put it. And remember, UPE research is still in its early stages.
Right.
But the potential is huge. It's like we've discovered this whole new language spoken by our cells.
And we're just starting to learn the vocabulary.
Exactly.
This is all so fascinating. But what are some of the biggest mysteries still surrounding UPE? What are scientists still trying to figure out?
One of the key challenges is pinpointing exactly which molecules are responsible for emitting those ultra-weak photons.
So we know it's tied to oxidative processes, but not the specifics.
Right. It's like we know the orchestra's playing.
But we're not sure which instruments are creating the specific sounds.
Exactly. Another big question is how UPE varies across different cell types, tissues, and organisms.
So what we learned from those HL60 cells in a lab dish might not apply to other types of cells.
Right. And then, of course, there's the question of how we can actually use UPE.
Yeah, like for diagnostics or monitoring disease.
Exactly. We need to develop more sensitive detection methods and figure out how to interpret the signals.
So much to explore. It sounds like we're just scratching the surface of what UPE can teach us.
I agree. It's a whole new frontier in biology.
It's really incredible to think that something as simple as light could hold so much information about our health.
I know. It's mind-blowing.
Well, on that note of awe and inspiration, I think we've reached the end of our deep dive into the world of ultra-weak photon emission.
It's been a fascinating journey.
It really has. A huge thank you to you for being our guide.
My pleasure. Thanks for having me.
And to our listeners, thank you for joining us. Remember, even the faintest light can lead to incredible breakthroughs. Keep those minds curious, and we'll catch you on our next deep dive. Thanks for listening today. 4 recurring narratives underlie every episode: boundary dissolution, adaptive complexity, embodied knowledge, and quantum-like uncertainty. These aren't just philosophical musings, but frameworks for understanding our modern world. We hope you continue exploring our other podcasts, responding to the content, and checking out our related articles at helioxpodcast.substack.com.